So I'm a medical oncologist by trade, but a very long time ago, about 20-something years ago, I did my PhD in a genetics laboratory. And that was a really sort of bizarre thing for an oncologist to do back in those days. But it's led into all sorts of sort of weird and random things in my career. And so one of the hats I also wear at the moment, as well as still seeing patients in the oncology clinic and doing various elements of oncology research, is I'm also cancer lead for one of the regional genomic laboratory hubs in England. And so what I'm going to try and do over the next sort of 20 minutes or so is just give you a sort of introduction to what genomics means, what it's all about, and how we're using it increasingly in the clinic these days. So to go back to the very beginning, we need to think about what DNA is. And essentially, DNA is the recipe for life. It's a very special molecule that makes up the genetic code within the body, and that instructs every cell on what to make, what to do, how to behave. And DNA is found in every cell of the human body, as well as most other organisms as well. And DNA essentially exists as long chains of molecules which are wound up and divided into structures called chromosomes. And those chromosomes are found in the nucleus, the sort of control hub of each cell. And we have 23 pairs of chromosomes. And we have pairs because we get one of each chromosome from our biological father and one from our biological mother. And we can pass on the material in the chromosomes to our children if we have them as well. So when we talk about the human genome, what we're talking about is the entire amount of genetic material that's found in any of our cells. And that includes areas of genetic code that make the recipe for certain proteins or characteristics in the body, and those are known as individual genes. So there are genes for hair colour, eye colour, all our physical characteristics. And then in between those genes, there are lots of other bits of DNA which we used to think didn't do anything at all, but we now know that's really important as well. So when people talk about looking at the whole human genome, they're looking at the sequence not just of the individual genes, but of all the genetic material in between. And we talk about genomics being the study of all the genetic material, but also how it works, and more broadly, the technologies we use to look at it. And the reason everyone's got really excited about genomics in the last sort of 10 to 15 years is because of the massive improvements in the technology available to look at the genetic code that previously was only available in very specialist science labs, but has now made it actually all the way into, the clinical labs we use in the NHS. We talk about a process called sequencing, which is when we actually analyze the DNA to see exactly what the sequence of letters is within it. And to put things into perspective, back when I did my PhD, I could set up a bit of sequencing on a machine, and it would take about 2 to 3 hours to look at a series of 150 letters in the genetic code, which was perfect because it gave us all time to go out to the pub for lunch, and then the results would be ready when we came back. And using exactly those techniques, scientists across the world did manage to use those techniques to sequence the entire genome of a human for the first time. But it took them 13 years and about £2 billion. These days, the techniques have moved on so far that we can look at a whole human genome in about 2 days, and it costs less than £1,000 to do it. So no time for pub lunches, but it does mean that sort of technology has moved from just research into routine use in hospitals. So this is, again, basically what we mean by sequencing of DNA. It means that we can take a sample of human tissue, blood cells, or a piece of tissue. We can ask our scientists to actually pull out the DNA molecule from that using sort of a special series of solutions. And then we can pop that into a very clever machine called a next-generation sequencer. And we can use that either to look at the whole genome or to look at very individual bits of it, the bits we're really interested in. We can ask it to look at specific genes or to look at for specific changes in genes. And what's amazing about these new machines is they can look at lots and different bits of the genetic code all at the same time. And that's why we can do it so much more quickly than we ever used to. And this is my favorite and utterly random fact about genomics, is that if you look at the sequence of the whole human genome and compare it to the whole genome of a chimpanzee, 96% of it will be exactly the same. If you compare it with a mouse, 90% of it will be exactly the same. And my best fact ever, if you compare it with a banana, there's 60% of it. We have the same genetic code of 60% of a banana, which I think is just brilliant. Okay, so what on earth has any of this got to do with cancer? Well, because cancer, ultimately, is a disease of the genome. It's something that develops when something goes wrong with that genetic code within the cells. And we know lots of things that can damage DNA and go on to cause cancer, such as the chemicals in cigarettes, too much ultraviolet light, certain viruses. But we also know we can inherit alterations in the genes from one of our parents. And as we get older as well, the techniques we use within our cells to reproduce DNA can start to get a bit dodgy, so that we can actually develop alterations in the DNA as we get older. And we often refer to these changes as mutations. Essentially, mutations are spelling mistakes in that genetic code within the DNA. And if you've got a spelling mistake, that can mean that there's an abnormality of the protein that recipe is telling the cell to make, or it can mean that the cell can't even make it at all. So essentially, when we have alterations in our DNA, we're getting faults in our instruction manual. And that can be a real issue if we're talking about the instructions that tell cells how to behave when they're dividing. So normally, we only make new cells in the body if we need them for a distinct purpose. For example, to repair wounds, to heal, when we're children, to grow. And normally that whole process is controlled really, really precisely by a whole family of proteins within the cells. But if there's a problem with that DNA, if there's a spelling mistake, then what can happen is if you get an alteration or a non-functioning protein that should be controlling cell division, you lose the stop signals and the cells will start multiplying uncontrollably, picking up changes, which means they'll behave badly, and that is essentially the basis of cancer forming. So when we're talking about genomic information in patients with breast cancer, most of the time we are talking about changes in the DNA sequence, the genetic material of the cancer cells, the changes that have caused those cells to behave abnormally. And that's becoming increasingly important because more and more treatments are being made that specifically target those changes within the cancer cells. And the important thing to realize when we're talking about genomic changes in the cancer cells is that these have developed a long time after the eggs or in a man, the sperm were produced, and therefore these cannot be passed on to your children. But the other type of genomic information in breast cancer patients we're also increasingly interested these days is the DNA sequence that you've always had, that you inherited from your parents. And this is alterations in that, what we know as the germline genome, that can actually affect somebody's risk of getting cancer in the first place. And it's those changes that can be carried on in the family. So we do two different types of genomic tests in oncology clinics. If we are talking to a patient about a risk of developing breast cancer, whether they have inherited an increased risk of getting breast cancer, then we want to look at their healthy genetic information. In particular, in breast cancer, we're talking here about wanting to test the BRCA1 and BRCA2 genes. We know people who have inherited an alteration in either of these genes have a much higher risk of getting breast cancer during their lifetime. They also have a higher risk of developing some other cancers. And there are now some specific drugs which have been produced absolutely to exploit the changes in the body systems that go along with those inherited breast cancer. But this does have implications for the family. The other types of tests are to look at the genetic sequence of the tumour. We call this a somatic genomic test. And this is generally done in order to look for particular alterations that are associated with sensitivities to certain new targeted drug agents. And again, these can't be inherited, so there aren't implications for the family. Now, in England, for the last sort of 7 years or so, all the genomic tests we do are essentially controlled by what was NHS England. I'm not entirely sure what it's going to be called next. And there are sort of similar authorities in Scotland and Wales. And what that means is we can't just do any genomic test we feel like doing. We can only do the ones that are funded by the NHS. We can only offer our patients the ones that are funded by the NHS because the laboratories aren't allowed to do them if we sort of go outside the guidelines. And I'm just showing here that these are the sort of thing we have to look at when we're assessing who is eligible for genomic tests. So if we're thinking about has a patient inherited a gene alteration that put them at higher risk of having breast cancer, we have a set list of criteria. Now, not everybody always sort of fits into criteria, and if we have patients where there's a really strong family history of breast cancer, but they don't meet any of these particular sort of criteria, we can still talk to our clinical genetics team and ask them to investigate further. There is another way, though, and that is we can also request testing for breast cancer susceptibility gene alterations in patients, even if they don't meet any of those other criteria, but if we think they might benefit from treatment with a special class of drug called a PARP inhibitor. And so patients with secondary breast cancer who might benefit from a PARP inhibitor, we can also offer genetic testing for breast cancer susceptibility genes, even if they don't meet any of those other things. One issue here I have to point out, though, is that this group of drugs is only proven to work in patients with HER2-negative breast cancer, and therefore we can't offer testing on this basis to patients with secondary cancer that is HER2-positive. If we do go down the line of recommending a breast cancer susceptibility gene test, it can be done by blood or saliva. And because it has implications for people beyond the patient we are talking to, we do a formal sort of consent process. We will ask the patients to sign a document to say we have discussed the implications of the test. And it does take about 6 to 8 weeks to get the result. If it's a positive result, we will then refer the patient to our colleagues in genetics for a full discussion about what that means and who to inform within the family. And so just to point out with the pathway, people often ask me, why does it take, you've just said you can do a whole genome in 2 days, why does it say 6 to 8 weeks to get a BRCA result back? And the reason is that although an awful lot of it is very automatic these days, there is still a lot of human hours involved, and nothing is reported out of the lab, particularly with this sort of inherited gene testing, without a lot of human hours checking and double-checking the results before it's released to us. So what about if we recommend tumour testing? Well, the way funding works for genomic tests means that we can only ask for testing of the tumour at a genomic level if it is likely to change the management of the patient. There are certain tumour-based genetic or genomic features that can provide eligibility for some of the new targeted drugs that have become available to us over recent years. But strictly speaking, if a patient isn't eligible for treatment with those particular drugs, they're not eligible for the test, okay? And that, I know that sometimes feels like a really, really hard decision, but these are expensive tests, and that is sort of how things are being done at the moment. So oncologists will discuss this sort of testing with patients who are potentially eligible for certain targeted therapies such as alpelisib and capivasertib. These are drugs that have been developed for patients with estrogen receptor positive, HER2 negative disease, and therefore currently I wouldn't expect your oncologist will be discussing these sorts of tests if you have triple negative or HER2 positive disease. It does, as I say, generally require a sample of cancer tissue. That can be from a biopsy. That can also be from the original breast tumour if a biopsy is not available. Because this sort of testing doesn't have implications for family members, we don't have to go through a formal consent process. Results generally take about 2 to 3 weeks, but sometimes we just can't get enough DNA out of those tissue samples, and it will be reported that the sample has not been able to be sequenced. And something we do need to point out here is that if one of these genomic alterations is found on tissue testing, it may well mean that we can offer patient treatment with a certain targeted therapy. Unfortunately, it doesn't guarantee that a patient will respond to that treatment. And even with the newest targeted agent, we are generally seeing response rates of about 50 to 60%. I've never seen 100% with anything. So it is about eligibility rather than guaranteeing response. There is a new type of genomic test, however, that's coming into play, which is potentially going to make all our lives a little bit easier. And that's a test called a liquid biopsy, or ctDNA. And this is still primarily a test used to look at the sequence within tumour cells. But it works on the basis that for most people with secondary cancer, there will be cancer cells in the circulation which are breaking down, and with modern technology, even the tiniest amounts of DNA from those cells can be worked up in order to be sequenced, just as in the same way we would sequence a tissue sample. One slightly complicated thing about this is that it's not just the tumour DNA that gets into these tests. It can also pick up DNA from healthy cells. And so sometimes when we look at results for these, we see sometimes very unexpectedly that the test is giving us hints that there might be an underlying alteration in the healthy cells. There could be, and certainly I've been in that really awkward situation where I've been telling a patient for the last 10 years they're not eligible for BRCA testing on the basis of the regulations we have to work with. And then out of the blue, I've done one of these and it's hinted that there is actually a BRCA mutation underlying. And I think that really highlights the fact that none of these tests are positive. There are reasons, sort of financial capacity, why we can't do BRCA tests on everyone. I think that will probably change in the future. But at the moment, we are in this situation where we can find BRCA mutations through this sort of test that we weren't expecting. Again, at the moment, we have very limited access to this. It's only routinely available for patients who might be eligible for a drug called elacestrant. And again, that is a drug for patients with ER-positive, HER2-negative advanced breast cancer who meet certain other criteria. But what about wider genomic testing? What about doing it in order to see if we can get patients into clinical trials of new drugs that I haven't mentioned? There's a lot of work going on this. NHS laboratories are being encouraged to report out more extensive genomic alterations beyond the ones I've mentioned. It's very much a work in progress. And certainly at the moment, it is still restricted to people who are already eligible for some of the genomic tests I've mentioned, but there is a lot of work going on to change that. There is also some situations which we could get access to the liquid biopsy testing within certain trials. And for example, there's the Target national trial, which lots of us here are recruiting to, which allows us to send off a liquid biopsy test on patients with secondary cancer where we feel that they might benefit from looking for options in terms of trials of new therapies. The last thing I just wanted to touch on is that there is a huge amount now. If you Google genomic tests for breast cancer, you will get pages and pages of adverts from companies I've never heard of. It's a really rapidly expanding market, and it's really, really problematic because the quality of the tests is incredibly variable. The quality of the reporting is incredibly variable. We've certainly had patients come to us saying they've been told they've got a BRCA mutation, and when we've looked and done proper testing for it, they're reporting the wrong thing. We've also had it the other way around, where patients have felt reassured by tests that haven't shown a BRCA mutation, but actually when you look in detail, they were only ever looking in very small parts of the genes and not doing the comprehensive testing we do today. These tests, of course, are promoted by people who are sales. Your staff, not clinicians. They won't know your particular situation, and they certainly won't be tied into what's available in terms of drugs within the NHS or with clinical trials as well. Another area that's causing issues at the moment is availability of ctDNA monitoring tests through commercial providers. This is an area those clinicians in the rooms, I'm sure, will agree, we're really interested in for its potential, but it's very much still at a research stage at the moment. And if any of my patients talk about having it, I always plead with them not to, because I won't have a clue what to do with the results at the moment. We're just not at that point. So I'm going to leave it there. I hope what I've got across to you is that we are increasingly using genomic information to help us give patients with breast cancer the best possible treatment. All the drugs that we are able to prescribe on the NHS now, they are covered by sort of the appropriate companion testing. Testing for breast cancer risk genes is changing the whole time, but it's still not, unfortunately, there for everyone. That may change in the future. And eligibility criteria have changed. So it might be something, if you ask for it about it some years ago, it might be that you are now eligible. So do have a chat with your oncologist about this. And I would please ask all of you to discuss with your oncologist before engaging with any straight-to-consumer genomic testing. I'm going to stop there, but I just have left there questions you might want to ask your friendly oncologist after today. If they can't ask them, tell them to give me a ring and I'll... Thanks very much.
Thank you very much, Ellen. So, don't forget to raise your hand if you have any questions. I do have some online. Is there anyone who was not yet? So I'll start with the ones online. So it says, in the NHS, looking at setting up in-house genomic testing labs to bring down the cost of procuring private testing down. Is the NHS looking up to do that? Absolutely. And is the cost likely to come down? And if that will happen, will it become more widely available?
I think absolutely. Absolutely, really good point. So since 2018, the NHS in England has done most of its tissue-based testing in-house. There are regional genomics laboratory hubs. What is still being done by commercial providers at the moment are the liquid biopsy tests, and they are pretty expensive as a result. But there is literally a sort of a bidding, I wasn't going to say war, I'm trying to think, battle, should we say, going on at the moment between the different laboratories about picking up that work and doing it within the NHS, and that absolutely should bring the price down. The downside is that it won't be as extensive. At the moment, we get reports of about 324 genes when we get one of these tests back, and the reality is we really don't need all that information at the moment. Equally, we don't want the NHS to invest in producing a test that's actually going to be outdated within 2 years' time. So there's got to be a sort of balance between the two, but it will definitely be cheaper when it comes through.
Let me just follow up on a question somebody asked me during the break about the criteria for access to genetics, so BRCA, PALB, PALB2 and so forth, that is still very restricted in the NHS. And since now there is a potential therapeutic implication, do you think in the very near future there will be an enlargement of criteria?
Yeah, so in terms of secondary breast cancer, we can refer anybody or undertake testing for BRCA mutations in anyone who might be eligible for treatment with a PARP inhibitor. So that is anybody with secondary breast cancer that's estrogen receptor positive, HER2 negative, who meets the sort of Cancer Drugs Fund criteria for treatment with a PARP inhibitor. So even if people haven't, particularly if they were diagnosed with primary breast cancer and told they didn't meet the criteria, they are likely now, if they've got the ER positive, HER2 negative type of breast cancer, to have access to BRCA mutation testing.
Triple negative?
Triple negative. Anybody, yes, so anyone, with HER2, so anyone with HER2 negative breast cancer, if they have triple negative, they're eligible as well. In terms of primary breast cancer, it is still more restricted, but the eligibility criteria are being added to every year, and there is sort of a definite sense that in the not too distant future, just anybody who has a diagnosis of breast cancer will be eligible for BRCA testing.
I'm HER2 triple positive, but I'm not ER positive.
Just put the mic closer.
Yeah. So in terms of the reasons we're doing genomic testing for routinely available treatments, because they are not treatments that are licensed in patients with HER2 positive disease. At the moment, we are not able to offer routine genomic testing for things like AKT, P10, P3CA, the sorts of things that have certain treatments linked in because they're only licensed for the HER2 negative, ER positive type of breast cancer. As I say, within Target trial, that is where you potentially be able to access liquid biopsy form of genomic testing if your oncologist felt that was a good idea, and particularly if they are looking at access to clinical trials. So the problem with private tests is what you're going to do with the results. And actually most of the development of the targeted agents isn't in the HER2 positive. There are a lot of drugs coming in that are very specific for other types, but not the HER2 positive. So it's unlikely at the moment to add a lot to an individual's care.
So just adding to that what I discussed yesterday. So don't ask the biomarker if you can't do anything with it, or if there is no treatment for it, right? Because it will only give you anxiety to know that your tumor has this alteration, and so what? I'm not going to do anything about it, right? And don't feel sad that the HER2 positive doesn't need these type of tests, because every treatment that you have is targeted. And is targeted against the most important alteration of your tumor, which is HER2. We just don't need these fancy tests to determine HER2. Any other question in the room? If not, there is an important one from a man, and I'm very surprised with the information he got. So I'm a man living with secondary breast cancer. In 2005, I asked about genetic testing as I have children, and I was told I wouldn't need testing as I have no family history.
So we've come a long way since then, and actually any male with breast cancer is eligible for BRCA testing these days, whether it's with primary or secondary disease.
Exactly. So it's very important for him and for everybody and all other men that you should demand to have a genetic testing, because there is a higher probability that there is an alteration, especially in BRCA2. Although still the majority of cases in men are not linked to genetic alterations, but every man is in the guidelines, every guideline I think, that should be tested.
And I think that really does highlight the changes in our understanding and the eligibility criteria of BRCA testing.
There you go.
Just going back to the commercial testing that can be done, is there no government code of conduct or guidelines? Is it just a free-for-all?
It's a bit of a Wild West, yeah,
Right.
Absolutely.
Yeah, and the problem is that there is even, if you look online, there are places where you can just send a little bit of your saliva and they will do whatever they say they do. I don't know exactly what it is. So you don't need blood, not even blood. Okay, and so you need to be very careful. There are, you didn't mention, and correctly so, but there are many things online that say that they will predict if a treatment will work for your cancer. Say they will kind of do this, I don't know if any of you have ever seen that when we have an infection, the lab does a testing of the most common antibiotics and then tells us which one has the highest probability of responding. And so they claim they can do the same with tumor and the different types of treatment. That has no evidence, it's not evidence-based. And so be very careful. I had several patients coming up with that and asking me to take decisions based on that, which cannot be done. So there is a question there, and then we have two troublemakers here. Oh, there was a question there. That's okay. Go ahead.
Now you've totally thrown me as to what I was going to say. Actually, I was really going to just make a very positive comment, which was thank you for such a clear presentation, Ellen, because I think we really, yeah, it was really good. especially differentiating between genetic and genomic testing and, what's relevant there. Because we really are in the Wild West here with this area, aren't we? Rather like the previous session that I was chairing, How to Spot a Fad with Nutrition, we've got all these people trying to make a swift buck out of people who are enormously vulnerable. And I just think it's disgraceful what's going on. But I will have a question as well, a little one, which is a very sort of general question that I was talking with Fatima about, which is the word mutation. Because I think some patients find that quite derogatory. And I once wrote a paper a couple of years ago about genetic testing, and the editors forced us to change everything where we said mutation to pathogenic gene alteration, not a mutation. And I just wonder, what's in a word here?
Yeah, no, you're absolutely right, Leslie, and there's been a lot of work actually showing. So some of the work I've been involved with, variant or alteration comes up, and that's certainly with all the sort of patient-facing information and information sheets. We make sure we try and use that language. I've used mutation today slightly deliberately because it is often what comes up on reports, and increasingly I'm finding patients are asking me for copies of their liquid biopsy reports, so I apologise for using it today. It was slightly deliberate, but absolutely we know that it's not a nice word, it's not always accurate as well. Sometimes we're talking about overexpression of genes, amplification, there's lots of other technical jargon we can use. But yeah, mutation is not a favourite of anyone's.
But perhaps, scientifically speaking, I think the result could say alteration. But then for us, scientifically, we need to know if there was a mutation, because it is different to have a overexpression or a mutation, and there are drugs that work for the mutations exclusively. So I think perhaps as an overall result, we could change the word to alteration. But for the details, we still have to use mutation. And I answer to Leslie that if it's a tumor that is a mutation, don't worry about it, it's just a tumor. We know the tumor is not nice. I think it is more problematic when we're talking about genetic mutations. Do you still have a question? Yes. Could she get the microphone, please?
Come in. So obviously all of this has changed a lot over a small number of years. So if somebody has had genetic testing in the past, but it was a number of years ago, should it be reconsideration for repeating genetic testing?
It's certainly, so we're talking about germline or BRCA testing. Yeah, it's a really good point, and it's definitely worth a discussion for lots of reasons, depending on when it was done. Firstly, back in my PhD days, registrar days, we didn't look at the whole of the BRCA genes, their long genes, the laboratories would only look at the sort of most common areas. The other issue is that as we have learnt more and more about this area, some gene alterations that in the past we didn't know a lot about or really what to tell patients about, we know a lot more about, and actually there are a lot now we can reassure patients that actually, yes, it's a spelling mistake, but there are no sort of consequences of it really, whereas others we've got a lot more information about them. So the other thing is that we test more than just BRCA1 and BRCA2 now, so it's definitely worth revisiting with your oncologist and asking them to chat to their geneticist about it.
Yeah, and I suppose on that as well, is there any plans for a national sort of database of what's been tested? Because quite often you'll get patients that come through, they might vaguely say, oh, I think I had some kind of test, but then you're not too sure, and it would be great to be able to know rather than duplication when we're thinking about resources in the future.
So there's a fantastic team at the ICR Institute in London who are doing a lot of look at this, and Claire Turnbull, who I don't know if any of you met, are just fabulous academic geneticists, how particularly now that we're doing tests, we sort of know where the tests are being done, how that information can be sort of captured and looked after. And I'm sure that's sort of what will happen going forward. looking backwards, it's far more difficult because there are lots of different genetics labs doing different tests. Locally, they even had their own hospital numbers for families, and everybody's records was in a big chest freezer, and and it didn't even sort of link to anything else in the hospital. So locally, different places, our team is very good at going back and looking, and when more tests come online, they've sort of like offering PAL-B2 to people who've previously tested negative for BRCA. So a lot of the regional labs are doing something locally, but you're absolutely right. Going forward, it would be wonderful to have a database.
Can I just add a little bit to what Ellen just said in terms of going back and retesting? Our local hubs are overwhelmed as it is, and so there may be a reluctance to retest. As a rule of finger, I think pre-2005, I think people will retest, but it will be done under R444 if you have secondary disease, if you have metastatic disease, because it may enable access to new drugs. But in terms of the full panel, which involves RAD51, all the things, that's not being redone for everyone who's already been through the system, because it wouldn't influence care now. Although the information may be helpful, we don't have a treatment that would change as a result of it. So the panel that women are now being offered, men and women are being offered now, is not the same as before, but we can't retest everyone who's been through the system unless it has direct clinical implications.
So there are a couple of questions online. I'm going to merge two of them they actually ask similar things. And we go back now to the genomics, so to the tumor, not to the genetics of the patient. So, for example, one is asking, is there a role of circulating tumor cells informing if a treatment can be relaxed or stopped, if there is a sustained long-term complete response? And the other question, very similar, is could the liquid biopsies save money for the NHS by testing patients who have a complete response and then maybe stop the treatment?
Yeah, these are huge research questions and ones we're really, really interested in. I have got a number of patients who have been on the same treatment for years, and every now and then we have a chat about, shall we stop it, shall we not, what shall we do? And there is a definite feeling that sort of genomic tests taken from circulating blood could be really, really helpful in guiding us make those difficult decisions, but it's still very much in the research phase at the moment. And again, this is why I would really counsel against doing that sort of thing privately at the moment, because until we've got the results from big research studies, we're really not going to know how to use that information.
And so just also a word of caution for those things that you can do on your own, online. There are a lot of companies that will test for circulating tumor cells, okay? So we don't know what to do with that. So if you come with that result, we will neither stop the treatment or change the treatment or do anything different, because that has never been proven what we should do about that. So, and it costs quite a lot of money also. Maybe a simple question, or it should be a simple answer, I think. What is the average turnaround time for a ESR1 test?
It's generally about 2 to 3 weeks, it tends to be about 15 days for us from sending it off.
So, anyone else in the room? So, no, don't send one. Speak on the microphone. Come on.
Sorry, it's a very personal one. Anyway, I've been told very recently that I've got the PIK3CA. Unfortunately, I didn't have time to speak to my oncologist about this, but in the report that he gave me says approximate variant alert frequency was 5% of reads. And based on that, I haven't got a clue what that means. Based on that, he reckons that changing medication will be of no use to me. So, yes.
Okay.
I am stable at the moment, yes, in what I am, in Ribo.
Remember, if it's working, don't change it.
Yeah, oh yes, I understand that. It's just that I don't understand because it also says this is a rare variant of uncertain significance.
Okay.
So I am like, okay, what does this mean?
So
Sorry.
try and
Sorry.
pick that apart. No, that's okay. So first of all, depending on sort of who has done the report, the PIK3CA variants we know can show or indicate sensitivity to a certain group of drugs that are targeted at the PIK3CA and other drugs in that sort of sequence you can get changes that are important in the sense, and we call these pathogenic alterations, but some of the laboratories will also report out sort of more minor changes that aren't necessarily, sort of provoking the cancerous cell. And actually, in terms of the drug, these matched drugs working, what we're looking for is alterations that are proven to be detrimental to the cell. So that's the first thing. So that's why if a genomic variant is reported, but it's a variant of unknown significance rather than a clear pathogenic variant, that is not something we'll usually use to guide treatment. But it does depend a lot. I always double check when I get liquid biopsy reports, I always go through and check the variants and check they've been classified correctly before I use them or ignore them. Variant allele frequency is essentially a measure of sort of what proportion of the cancer-based genomic material is showing the alteration, and it can vary hugely in terms of based on exactly what technology was used to do the test, exactly what reporting system and software they've used, whether it was tissue, whether it's liquid biopsy. So generally speaking, we don't have any absolute sort of guidelines in terms of using VAF to guide our treatment.
But it's a bit strange that they say a variant of unknown significance in the tumor. I've never seen a report like that.
And I wonder what sort of...
It's very strange. But if you have the report, maybe Ellen in the break can have a look.
Even the commercial reports will give you a big list
Yeah.
of the...
So I think we have to stop. But there is a question online that maybe gives you an idea for next year. I didn't ask on purpose because you could talk 20 minutes about it. It says, what is the role of AI in genomics? Maybe next year.